Okay. PowerPoint set 3 from the biochemistry unit, carbon and diversity of life. If you can't tell already, each of these is review from chemistry and then content that we have kind of already covered earlier in the year, bringing biochem together. Carbon. So we've talked about water already. Carbon is the backbone of a lot of living organisms because we're made up of carbon molecules. Proteins, DNA, carbohydrates, they are all composed of carbon compounds. There is a reason carbon is so important. It's because it makes up all those living things, and the study of it is organic chemistry. Most organic compounds are a string of carbons with oxygen, nitrogen, and phosphorus linked up in some way, but it's usually just this long row of carbons. Thinking back to this is review. The Miller-Urey experiment basically shows that we can go from not having living stuff to having living stuff. Think back to like the beginning of the planet. Basically, he put water, methane, ammonia, and hydrogen in this glass tube, which we predicted were the compounds that existed before life, and basically linked up some electrodes to that. The liquid water was heated to create vapor. Sparks were fired between the electrodes to simulate lightning through the atmosphere and the water vapor. And then the atmosphere was cooled again, so the water would trickle back into the environment, and this continued. Within a day, it turned pink, and within 2 weeks, 10 to 15% of the carbon, or 10 to 15% of what was in there, had become carbon compounds just by lightning basically shocking those different compounds together, which is really cool. They forms amino acids, glycine, proteins, but the most common 20 amino acids were formed, which is essentially how we get living things eventually. Carbon has 4 valence electrons, which basically means that it can bond with a variety of atoms, allowing it to make really large, complex molecules, because it can make 4 bonds that radiate out from it. Usually it's going to be a string of carbons, right? So that's a bond on the left and right side, and then the top and the bottom could be a carbon 2, or it could be a hydrogen or something else. We can also see 2 carbons creating a double bond between them, and only having one other or 2 other bonds, and that shape is important, either linear with a double bond or tetrahedral with 4 bonds, because that determines its reactivity or what it does. You don't need to memorize these, but these are all different types of carbon bonds that we see, right? A single carbon with 4 hydrogens, single bonds between carbons, double bonds between carbons. They all have important things. Methane, ethane, gas, ethylene, gas, all important. And again, we look at all of these different carbon, we call them skeletons, it's carbon linked with carbon, that can make all sorts of things. These are all different gases, benzene, butene, propane, ethane, gases, but they're all carbons and just hydrogens. Hydrocarbons are what we saw prior, just hydrogen and carbon. Most organic molecules are mostly hydrocarbons. Fats and lipids are basically just hydrogen and carbons, and they are very large molecules, and they release a lot of energy, which is why when I burn fat or I utilize fat, it's going to release a lot of energy for me. Fats look like this. The fat molecule is just basically hydrogens and carbons linked together. Isomers are going to be same number of carbons and hydrogens, different structure. If you think about trans fats that we have, there's two types of fats, right, that we see, and people talk about like the good fat versus the bad fat. Same amount of carbons and hydrogens, just a different structure, which means they can be broken down in a different way. A structural isomer, same number of carbons and hydrogens, just different arrangement. You don't really need to know this. Cis trans isomers are exactly the same structure, just a flipped portion. Again, this is like different types of the same number of carbons and hydrogens, just different arrangement makes different atoms or compounds. Again, this is just for your edification if you feel like you want it. Again, we can make molecules that are basically mirror imaging. It's a type of isomer. It makes them do different things, but they have the same number of carbons and hydrogens. Functional groups are going to be what makes these compounds differ. Basically, we have this one long chain of carbons, and at the end, it has some weird thing attached. So if you look at functional groups are mostly in organic chemistry, because usually it's just a long chain of carbons, and then this little thing that attaches to the end of the compound that makes it do something special. So these are tons of examples of functional groups. You don't need to know these. It's just showing that there's all these different ones. So for example, chain of carbons, an OH functional group is called a hydroxyl functional group. It can bond with water. It is sort of polar because it's got an oxygen and a hydrogen. It gives it special properties. Ketones or a carbonyl group is going to make ketones, which are things we see in our body. Both acetone and propanol have a carbonyl functional group to it. Carboxyl is going to be COOH. That is attached, whenever that's attached to the compound, it acts as an acid. Amino and H2 is going to be an attachment that we see. Other ones, you don't need to know these. The idea is that they show up. Phosphates, methyls. So something that's important is that in biology, we see that we have this long chain of oxygens and phosphoruses, and then we have this functional group, adenosine, on it, which gives ATP this very special property. It's a very strong bond, so it releases a ton of energy when the phosphate or when it's broken apart, which is why ATP is the source for a lot of our energy. Final things to think about, or to summarize, the versatility of carbon allows us to have a lot of diverse organic molecules, therefore a lot of diversity in life. So if we think about all of the living things that we see, it's because carbon can be arranged in so many different ways. Variation at the molecular level allows us to see diversity at a more visual level. So our diversity, the diversity within humans, right? Our diversity in skin color, hair, how we behave, goes back to the structural diversity of our molecules. And so we can't have the bio definition of diversity without the chem definition of carbon diversity. This is just something to think about as you're moving forward, and that is that. Science.